Yamina here. Welcome to the Dr. GPCR podcast. The path to science is rarely linear, and today's guest, Catherine Demery, proves that. From pharmacy school to industry, the NIH, and now her PhD at the University of Michigan, Catherine has shaped a purpose driven career around one of the most urgent crises of our time, the opioid epidemic. In our conversation, she opens up about how personal loss fuels her research, how she turned blind spots into strength, and how her work connects the lab bench to real world impact. Before we dive in, did you know that Terry's Corner is now live? This week, Dr. Terry Kinakin unpacks competitive versus non competitive antagonism, how to tell them apart through receptor binding and kinetics, how they reshape dose response curves, and the misconceptions that trip up even the most seasoned scientists. Terry's Corner is the only expert led, on demand pharmacology corner where Dr. Kinakin breaks down complex concepts into practical insights. Each week, we release a new course and host live monthly AMAs with Terry himself. A big thank you as well to our partner Celtaris Research, helping accelerate GPCR drug discovery with their innovative chemical biology tools. And if you want to sharpen your edge as a GPCR scientist, become a Dr. GPCR Premium member today. You'll unlock the full edition of the weekly news that includes curated jobs, curated publications, and over 4,000 minutes of expert-led courses on GPCR pharmacology and drug discovery. Check out the links in the description. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR. And this afternoon, I'm very excited to have Catherine Demery with me on the podcast. Catherine, welcome.
Thank you.
So excited to have you on. We've postponed this recording a couple of times, but I'm excited that we're finally doing this.
Me too. Thank you.
Right, so thank you. Let's start at the beginning. Can you please introduce yourself? Tell us who you are and what do you do?
Sure. So my name is Catherine Demery. I am a third-year PhD candidate at the University of Michigan Medical School in Ann Arbor, Michigan. So I primarily study opioid overdose in mouse models with a focus on fentanyl-induced overdose and respiratory depression.
That is awesome. What got you into science in general? And we'll walk through time to get you to your third-year PhD student position right now. But as a child, were you always interested in science, or you kind of stumbled upon it?
Sure. So I would say I definitely had an interest in science from a young age. I always did like the science fair projects, and really enjoyed tinkering and doing things like that with my dad down in his workshop. And I would say that, continued through high school. I was actually president of the Science National Honor Society for a couple of years in high school. And when I got to undergrad, I decided to major in biochemistry. And I really actually focused most of my undergrad around becoming a pharmacist. So I didn't actually do any research in undergrad. I prepared for the PCAT. And I took the PCAT, and I actually ended up getting into U of M pharmacy school. But then I did 2 weeks worth of shadows for different types of pharmacist jobs. And I just had this identity crisis kind of, because I think I realized in that moment that I didn't want to be a pharmacist, but I had tailored 4 years of my life to doing so, and had just kind of achieved what I thought was going to be this huge milestone in my life, and now I wasn't sure if I wanted it anymore, and it wasn't cheap, so I ended up deferring, just to give myself a little bit more time to think about it. And I kind of realized that I think I liked the pharmacy science from the other side, from the more research driven aspects instead of the clinical aspects. So I actually wasn't quite sure what to do with that. I was feeling a little still kind of shook after having, made this big choice to not pursue pharmacy school. So I ended up getting a job at a contract research organization in Ann Arbor, Michigan, which was called Avamine at the time. It's now bought by Element Materials Sciences. And I was an analytical chemist. So I did a lot of project was different, which was something that was really cool about that CRO, because a lot of them, you're doing the same assay over and over and over. So I really appreciated that aspect of the job. I got to work on I got to design some really cool, tests for preclinical trials and things like that. So I worked there for about 2 years, and I kind of felt like I gained the lab skills that I could, and the ways of thinking that I could up until a certain point. And that was long enough for me to realize that I wanted to go back into the more biological sciences, instead of the more harder analytical chem. So I decided to go pursue a master's, and I was really interested in pharmacogenomics. It was just something that I niche that I really, really found interesting. So I specifically joined a master's program at Manchester University in pharmacogenomics, and I had a really great experience. I approached it with a much different attitude than I had approached most of undergrad. I really wanted to make the absolute most out of my experience there. And I just had a different attitude than I had a few years ago about school and applying myself. And about halfway through the master's program, I had like a light bulb moment where I felt like for the first time in my life, I like understood why people pursued a PhD. And up until then, it was kind of lost on me, just like, so much extra school. And I don't know. But we got to choose our own research project in our master's. And so I chose to do like a review on genetic variation in alcoholism and opioid addiction susceptibility. And I just was so in love with that project. I mean, I was staying up, super late. I was excited to like work on this, till 2 or 3 a.m. And that was kind of when I was like, oh, I get it. People are really passionate and really happy about what they do. And that's why they go on to like pick an area of science to like dedicate their life to. And then I decided that like, this is it. I want to have a job where I'm so excited about what I do that I like stay up late working on my, my research. So I had, like I said, I hadn't done any research in undergrad. I had a couple years in industry under my belt, but not, grad school is pretty hard to get into these days, especially R01 universities like U of M. And I already lived in Ann Arbor, so I really wanted to get into U of M, and I knew that I wasn't quite at I would have been competitive, I think, with my master's research experience, but maybe not a shoe-in. And I was really fortunate to land a job as a research assistant at the NIH after graduating my master's. And so that was actually the Detroit Medical Center, up until I think 2 years ago, used to host a branch of the National Institutes for Child Health and Development. And that's because Detroit has one of the highest rates of preterm birth in the country. And so they housed this particular branch there in order to, be able to study this using real life patient samples there. And so I worked there for a year under Dr. Nardi Gomez Lopez. And, I felt like I learned just as much as I did in my master's in that 1 year working at the NICHD. It really catapulted my career. And I'm really grateful for that opportunity.
That is fantastic. Hey, we need to track back, because I think that this one thought I had when you mentioned pharmacy, typically our guests on the podcast, either they've always wanted to go into research, which is very rare. We have the med students, the group in which I am as well, but then we have the pharmacist, and Arthur Christopoulos actually is a pharmacist. I think he even still has his license. The reason why this came into mind, because our podcast is broadcasted on all sorts of podcasting platforms, including Spotify. And I just got the 2024 recap, and Arthur's episode was the most listened to podcast. So hence the connection between you, pharmacy podcast, and Arthur. But I found it very interesting that you decided to, after undergrad, to go onto the job market and work at a CRO. Was it because you felt like you didn't have enough research experience and you wanted to get a sense of what does it look like to work in the lab? or were there any other, thoughts around just having quote unquote a real job experience?
Yes, excellent question. So more so the latter. I would say that I honestly I felt pretty like lost for a bit there after having kind of had this big arc with the pharmacy school, and then choosing not to pursue that. I just I was working as an assistant banquet coordinator in downtown Ann Arbor, and I was like applying for every technician job I could find. I just at that point I felt like I needed some sort of scientific job to feel better about having sacrificed the pharmacy opportunity. And I actually, out of all of the jobs that I applied for, I got the best one. So I was really happy about that. This was, a real chemist position, not, so, a little bit more responsibility than a technician role. But, honestly, I just felt thrilled to like be using my degree and be in a lab setting. And that was really enough for me right then. Was it certainly had not crossed my mind to apply for graduate school right then. I really just felt like I needed something to keep my scientific self-esteem up, and just kind of to get my foot in the door, so to speak. It really is true that like landing your first job is by far the hardest, and it kind of was, but that was truthfully why I did it. It wasn't really with much foresight for like a couple years down the road. It was mostly just like I need to be back in the lab. I want to feel like I'm using this bachelor's degree that I just got, and I know that the longer I go without it, the harder it's going to be for me to break back in. So yeah.
And I really like that you ended up working at a company, at a CRO. I think it's a completely different setting than working in a research lab, in an academic research lab, compared to even working at a biotech setting. And a lot of people in the audience may not know the nuances between this type of work, but I think working at a CRO, and please feel free to chime in on this, gets you the discipline, the experience, but also the concept of this is for a client that's paying for this, and this needs to be done well and on time.
Yes. Yeah, you hit on that perfectly. Yes, it is a very, very different world from academia. And I would definitely do it in the order that I did, because I agree that, yeah, the CRO was very, deadlines were really, really intense. I mean, you were expected to meet those deadlines pretty much no matter what. Because yeah, there's a whole company on the other end who's waiting for this deliverable. It's not just a matter of like, oh, we might not publish this as soon as we wanted to. No, it can be, really high stakes for the companies that are hiring us. And so yeah, the deliverables deliverables are kind of the end all be all, and deadlines. And I would say sometimes that quality suffers before the deadline suffers, and that's not always the best thing. And that's not to say that that's the case in most cases. I just say in isolated instances I've observed that to happen. But it's just a much different environment. Like you touched about the rigidity, and so a lot of the work that I did at the CRO was GMP, good manufacturing practices, or GLP, good laboratory practices. And so for these, you have to have really stringent documentation. You have to have, you have to have somebody observe a part of, what you're doing. I mean, it's really, really structured. And really I always used to joke that you spend just as long documenting what you did as actually doing it, which was certainly true. But then, to years later kind of be in academia, even when I was at the NIH and, taking these like obsessively long protocol notes, and, they're like, you don't need to do that. I'm like, what do you mean I don't need to do that? They're like, it's fine. Like, just write down that you like followed what you did like 3 days ago. And I was like, oh, are you kidding me? Like, is this right? Like, I don't know. It was just crazy because I had started out there. I was like, wow. So I'm glad I went in that direction. I think it's a lot easier to adapt to looser restrictions than it would have been if I had, already had all of these laboratory habits that I then had to,
develop then, and then actually unlearn some of the laxed
exactly.
So absolutely. So, and maybe if you can tell us a little bit more about what are the things that you learned at the CRO that you're still using in the lab today and doing your PhD?
Sure. So a lot of the really basic lab skills, I did a few labs in undergrad, but again, without the research focus, I just kind of did the minimum. And a lot of them were like more like organic synthesis based classes. So not really like pipetting and things, very normal techniques that you would use in almost any wet lab. So I felt like I just got like all of the basics down. And then I also did a lot of mass spec, and different types of mass spec and HPLC, and a lot of analytical techniques that I don't use now, but I think it's still really, really helpful to understand how all of those work, because lots of papers, or even sometimes we use CROs to gather like pharmacokinetic data. And so just having an understanding of how all of that is collected is really useful. Also, just basic proper ways to do things. Like it's better to prepare every standard individually than to do serial dilution, because you could just serial dilute one mistake. And so kind of good practices like that are forced in those industry CGMP, CGLP settings, then stay with you and are okay, well, this is how it should be done. And so a lot of those habits, and then I would say just the really basic lab skills.
I love it. I love it. And I'm assuming then once you finished your master's and you ended up at NIH, and I didn't know actually that NIH had a separate lab there in Michigan. I actually was also at NIH, but in Bethesda in Maryland for quite some time. But what led you to that specific lab? Was it the scientific interest in per se, or did that experience at NIH as a research assistant give you the time to think about, okay, what next for me?
Yes, absolutely. And I am curious to hear about your time at the NIH after I give my answer for that. But so I knew after being in my master's, I knew that I wanted to go to grad school. I knew I wanted to be at U of M. And I was, happy to apply elsewhere, but I knew that I was gunning for U of M again. I just had a great undergrad experience there. And I mean, they're, if you're going to be in Michigan, they're the best. Sorry. So I knew that I needed something where I could get, a lot of different research experience. And so I was just applying to all sorts of open positions. And I saw this for the perinatology research branch at Wayne. And I, there was a lot of confusing parts there because it's held at the DMC, but staffed by Wayne, and then is a part of the NICHD. So I really had no clue what I was truly applying for. I wasn't, I didn't really understand that it was in fact a branch of the NICHD at that point. I just thought it was a research branch at Wayne. And Yamina, I Googled what perinatology was. So I had, I'm just being honest. So I had no idea what, I think at that point I knew I'm somebody who can get easily excited about any type of science. And I knew my heart was really set on doing research into opioid abuse specifically, but I was happy to take whatever side rails I had to do in order to get there. And I am really glad that it was something so off topic from what I had studied before, because I had always considered immunology to be one of my weaker subjects. And most of the research that I did there was focused on how, the adaptive immune system changes in pregnancy to accommodate, the semi-graft fetus. And so it really forced me to kind of patch up all of my immunology holes and then apply them. And I really just came away from that job with just a whole new appreciation for immunology and for pregnancy. As a woman, I had always thought it was cool that, we could grow a baby inside of us, but I had never thought about it on such a molecular level, how our T cells are changing and all of, it was really, really fascinating and kind of just eye-opening to a part of the world that I don't think I would have, put that much thought into ever again. So I really enjoyed doing that little detour.
I love it. Yeah. Unless you are pregnant, you don't think about all of these details. And if you're pregnant and a scientist, then you think about all of these details.
Exactly.
Which can be very important. And I think there's a lot of research that needs to be done. I don't think we still understand some of the intricacies. I've always been fascinated, speaking of pregnancy, as to, they give you a due date, but then for some reason, the baby decides to come early or later, but what is the determinant? What are the molecular determinants that will determine, okay, now is the time we're inducing labor? Labor is starting now.
Right. Yeah. Labor is super inflammatory. And yes, there's, preterm birth can like arise in so many different ways. And a lot of them to this day are still, idiosyncratic, and we don't understand. We do understand how some preterm birth happens. And we're learning more and more, especially Dr. Gomez Lopez's group has just done abundant work on all of this, of course. But yeah, there's still like a large percentage that are idiopathic, and we really need to try to figure out, because that's a huge cause of I think it's for sure the leading cause of neonatal morbidity worldwide. So that's obviously a really important thing. So I'm curious to hear about what you worked on at the NIH.
So at NIH, I actually, I told this story for those who listen to the podcast regularly. I apologize.
Sorry.
But basically, I ended up at NIH thanks to a collaboration. I was at a conference, and I was presenting a poster on signaling really basic molecular pharmacology of chemokine receptors. There was this one chemokine receptor, which is one of my favorites. It's called CXCR3. It has different splice variants. And I didn't want to work on this. I was more interested in a heterodimer of CXCR3 with another chemokine receptor, which is an atypical one. But anyway, long story short, I put together this poster. I created all these biosensors to measure the signaling activation of the 3 different splice variants in response to 4 or 5 different chemokines. And someone introduced me to a PhD student at the time who said, oh, you should talk to Ian. Ian would love to collaborate with you. And that fast forward 9 years, we finally ended up publishing the collaborative paper that we worked on together. But the introduction to Ian at this conference got me introduced to a PI at NIH. And when I was looking for my next opportunity, that PI particularly introduced me to my boss at NIAID. So it was basically a, telephone type of thing of, oh, I'm looking for a postdoc, I'm looking for a trainee. And I went and interviewed, and that was pretty much it. So I moved from New York City to NIH, and it was a very interesting time because for those listening, if you've never worked in government, it's a whole new world. It's very different. Things are tracked differently. Well, you do still do your research, but one interesting thing I found is the fiscal year ends in September. So from September 1st to October 1st, you can't spend. So you have to plan ahead and order all your reagents, or you have to justify everything, which is a completely different way of going around. But what I really liked at NIH is that I was able to collaborate with a bunch of people. You needed something, you would find the person to talk to get help. So it was a fun period.
That's awesome. And you said you were there for 10 to 12? Sorry if I missed it.
So I was there for about 3 years.
About 3 years. Oh, I'm so sorry. I don't know where I got those numbers from.
No. The 10 to 20. Well, actually, I spent about 20 years in academia and not in government before moving to industry.
Oh, okay.
Yeah. As an undergrad, I did biochemistry as well. I did a master's, actually a bachelor's, a master's, and a PhD in biochemistry. And during my second year as an undergrad, I got the opportunity to work in a lab, and I actually worked 9 years in the same lab. I did a summer internship, a master's, and a PhD in the same lab.
Oh, wow.
So I was a local patriot when it came to that lab, but the minute I stepped into that lab, I felt like I was in the right context. I had the right support. I really loved the project, but at the same time, the lab was empty when I started. There were bills around. There was the PI and maybe a laptop. No, not even a phone, I think.
Wow.
So I got to witness that building from the ground up lab.
I'm sure that was an interesting and at times difficult experience.
Yes. When you needed to look for a buffer that had ATP in order to do your molecular biology reaction, because we didn't have ATP in the lab at the time, and it was too late and everybody had left on the floor. You learn how to improvise for sure. Really so cool. Let's get back to you. I'm curious to hear about, so clearly with time you got more research experience, and you decided that you realized that you're fascinated with science, with research, and you decided to do a PhD. But what led you to this particular PhD?
Sure. So I have always had, I've always, just to be super candid, been really passionate and somewhat sensitive to people who struggle with opioid abuse. I just, I had a few friends when I was younger in high school and a little bit past that, who, became addicted and really sadly, many of whom actually, passed away as a result of an overdose. And so that certainly shaped my interests and shaped my passions as a scientist. So I had some personal reasons for, pursuing this, but I feel like most of the time when people are really specific about, an area in science that they want to pursue ardently, that normally there's like a personal connection there somehow. And so, there certainly was for me too. And I kind of think that because of my, more personal experience or exposure to the other side of it, that I think that it makes me a particularly good person to research this at the bench, because I kind of have an understanding of what we're actually trying to achieve, and what we're the people that we are hoping to help. And I think it's important to kind of to stay aware of that. As scientists, sometimes it can be really easy to get stuck in the minutia of our receptor and the coupling interactions, and on that particular level there's so much to explore and to think about, that sometimes sometimes I think that people can lose sight of the really big picture, the zoomed out purpose of their research. And so that purpose is like always really at the forefront of my mind, and it really keeps me motivated and passionate when you get your PhD lulls or burnouts, as they say.
No, I agree with you, and I think and to your point, it's very difficult to keep in mind that 30,000 feet view when you're working on figuring out how many microliters or how many micrograms of DNA should I mix for my biosensors to get the response. But it also allows you to, be able to do something that's really challenging, is be able to go from the minutia to the, very close to the actual micrograms of DNA to the 30,000 feet view. And if you're able to do both of those, to go down into details, but also come up and try and understand, okay, why am I doing this and how does this affect my higher purpose, which is to help people or better understand a mechanism and then develop drugs, for example, that allows you to, one, keep things and put things into perspective, and at the same time, keep you motivated.
Yes, absolutely. And I guess at some point I should specify that I work in the lab of Dr. Drong Trainer, who's been like a pioneer in the field of opioid pharmacology for many, many years. And I'm also co-PI'd by Dr. Jessica Anand. So I felt like at some point I should definitely throw their names out there. So people could find more information about their research if they wanted to. So.
Absolutely. And in the guest form, there'll be an opportunity, if you haven't filled it out yet, to put in the lab website and anything that you'd like to share with us, because I think it's important. One thing that we've been focused on at Dr. GPCR is to bring people together. And not everybody gets to go to the Gordon conference. Not everybody gets to go to the GPCR retreat or any other conference. These are the two ones on my top favorite list. But we need to have a platform where we can collaborate and we can meet and exchange. And I think the podcast is one of those ways of shining a light on these scientists that don't necessarily always have the stage, but at the same time it's important for us to get a sense of who does what and where can we learn from each other.
Yeah, no, absolutely. I think that's really wonderful. And yeah, I've looked at a lot of I've listened to some episodes, but I've definitely scrolled through all of the people that you've had and certainly recognized plenty of names there. So that was pretty cool.
Awesome. And we're always looking for names for people to join the podcast, no matter what they work on, as long as it's related to GPCRs, no matter where they are, no matter what their level is. And I'm very excited, to be able to have you on as a PhD student. Because as I was mentioning before we hit record, and I want to give a shout out to our Dr. GPCR team, for finding a new way to get the students onto the podcast. It's been a fantastic, approach. So if you're about to publish, you have published your work, come join us and let's have a chat about what you've done. All right, that was my publicity plug for the podcast here. So let's get back to you, Catherine. You decided to do a PhD when you were looking. Well, obviously the topic was important for you, and you wanted to stay at in Michigan, obviously University of Michigan. But how did you approach finding this PhD position? Did you apply to other positions, or you knew from day one, this is where I want to do my research?
So I will level with you that I only applied to U of M for family reasons. I needed to stay local. And my plan was that if I didn't get into U of M, well, I knew I would get in the year after. If I didn't get in the first year I applied, I was pretty confident that I would get in the following year. And I had decided that it was worth it to me to wait a year if I needed to and go to U of M versus not waiting a year and, moving to Lansing or, to Detroit, or, I just thought, what's after, having taken however many years off since undergrad already, like what's one more year? So I know that's really atypical, and that's not necessarily what I recommend for everybody else. It's just, for me, that's the honest truth, was that I knew I was a competitive candidate, and that U of M was where I wanted to go. I had done like a preview program there where which is a really cool opportunity where you get to interview with like your top 2 faculty. You get to do a tour of the labs, and so I had the opportunity to interview with my faculty that I was interested in, go get a tour of the labs, kind of meet and chat with everybody, and I was familiar with the culture at U of M from having gone here for undergrad, but I would say that the grad school culture is a lot better. Even it's much less competitive, which is not what I had expected for, such a high caliber university, but it's really supportive. It's really not, it's not cutthroat at all. People really want it, like students really want their peers to succeed. And it just overall does not have, the competitive vibe that I've certainly heard of at other schools.
I love it. I do remember. So I studied in Montreal, and after high school, you have to go for 2 years to what they call college, which is cégep. And that's the 2 years before you apply to go to university, to undergrad. And so at that school, that says the most competitive school, everybody wants to go to med school. And I've heard of horror stories where people would hide books.
Oh man!
So that you can't find them in the library. This was a time where you can Google things. But at the same time, that's not cool, hiding books.
Not cool. No.
Others.
No, not cool at all. Do you mind if I take a couple minutes to talk a little bit about the paper that I just published?
Absolutely.
That you found me through? Cool.
Yes. Before we go there, I have one other question.
Oh yeah.
So
absolutely.
The GPCR aspect, when you realize that opioids are related to opioid receptors, how was that realization, where you went from 30,000 feet view to the molecular aspect of it? And then we can talk about obviously your research.
That's a really interesting question. I like it. So I would say it happened slowly. I would say that in my master's, doing the the review on opioid addiction susceptibility, I certainly got familiar with the mu-opioid receptor encoded by OPRM1 and the different variants that are pretty commonly discussed, like the A11G. And also, other dopamine, DRD2, various different genes that could be involved in that. And so then I kind of came to associate, okay, I knew then that all abused opioids bound to the mu-opioid receptor, which is, induces all of like everything from, the clinically beneficial effects of analgesia or pain relief, also euphoria, and then also the potentially lethal effects of respiratory depression. And then, that was the only opioid receptor I thought about until my PhD. And then now, I'm surrounded by people who work primarily on the delta opioid receptor, or, some people are working on kappa, and, not too many people work on the nociceptin receptor yet. I know that's, has been kind of the understudied stepchild of the opioid receptor family. But yeah, I think I really only considered the mu-opioid receptor up until maybe even a little bit into my graduate school, I would think. And now, I think about them in relation to the whole family of receptors. And for a lot of other compounds, particularly in endogenous opioids, they're dirty. They bind to not, to several different ones, some more than others, but they can bind to, almost all of them, depending on which endogenous ligand you're talking about. So then it's, now I really feel like I have an appreciation for it them as a family of receptors, and not just the one that, I cared about for longer. So I would say it's been a slow journey.
I love it. I love it. And let's talk a little bit about the paper that you published. And I'd love to hear more about the context of this publication with your PhD thesis overall.
Oh, absolutely. Thank you so much. So I recently published a paper, submitted to JPET, hopefully will be published soon, looking at how xylazine, which is a veterinary anesthetic, impacts the risk of fentanyl overdose. So within the United States, in the past couple of years, there's been a really big increase in the use of xylazine to cut fentanyl, illicitly manufactured fentanyl. And this is bad because it's in the place of less harmful adulterants like mannitol, pretty much like inert stuff that they're just, cutting the fentanyl with. And then here now instead we're using a sedative to cut the fentanyl with. And so obviously that can have a whole host of issues. And xylazine acts through the alpha-2 adrenergic receptor. So primarily through the alpha-2 autoreceptors in the central nervous system. It causes a decrease in sympathetic tone, so lowers release of norepinephrine, epinephrine. And so just overall, causes overall sedation. And so some groups have shown in mice that xylazine potentiates the lethality of fentanyl, which obviously, then has implications for human overdose. But nobody had specifically, shown the clinical or, the mechanism behind that. And so we wondered whether it was, because xylazine worsens fentanyl-induced respiratory depression, or could it be other effects of xylazine, such as those on heart rate. And so, I looked into that and basically used, administered mice fentanyl, xylazine, or a combination at multiple doses, and using whole body plasmography and pulse oximetry in mice. I showed that basically xylazine can decrease breathing rate below that of fentanyl, which is very scary. However, the xylazine does it in a different way. If we think about, one breath is breathing in and breathing out, fentanyl affects the process of inhalation. So it drastically, it lengthens the time that you're breathing in, but it really decreases the flow. So, you kind of take in these really slow, shallow breaths, exactly. And xylazine, on the other hand, causes a massive increase in the length of the time that you're breathing out. So if you can imagine just really slowly, just breathing out as slow as possible. And so over time, that means you take fewer breaths in a given amount of time. And so it really, I was able to show that fentanyl and xylazine produce, different effects that both decrease breathing rate. And surprisingly, when they were added in combination, the breathing rate depression was not worse than that of xylazine alone at the various doses, which was really interesting and somewhat, relieving. So we also, though, were able to show that for the changes that fentanyl causes in the process of inhalation, that xylazine does exacerbate those. And so that, is not good, of course. But fentanyl doesn't really have an effect on the changes that xylazine produces alone.
Interesting. So there's no additive effect between the two from an inhalation perspective.
From an inhalation perspective, yes, but not from an exhalation perspective. And overall, that didn't translate to an additive effect on breathing rate.
Okay.
And so that was really interesting. I also looked at oxygen saturation, since, in an overdose, what ends up triggering cardiorespiratory arrest is, a lack of adequate oxygen in the heart. And so The one caveat that I'll make here is that mice are more resistant to changes in, in oxygen saturation than humans are, because they have a really high metabolic rate, and so they can adapt pretty quickly to those things compared to us. And so basically, if we see a change in mice, we're certainly going to see one in humans, but just because we didn't see one in mice doesn't mean that we wouldn't see one in humans. So I always just want to caveat that. But I actually was, surprised to see that at the doses that we used, and in the system that we had, that the addition of xylazine didn't worsen the maximum hypoxic effect of fentanyl. And so that was surprising because, xylazine had caused such a bigger decrease in breathing rate. And what I ended up figuring out, I think that explains that, is that, fentanyl induces apneas. Apneas have been shown across species to decrease oxygen saturation. And that kind of ends up being a pivotal point in a different manuscript that I'm working on now too. Xylazine on its own doesn't induce apneas. Even though the breathing rate is slower, it's much more regular. And so, I was not able to detect any apneas. And when, the two drugs were combined, the mice showed the same number of apneas as with fentanyl alone. So, that makes a strong case for a correlation between, the number of apneas and, loss in blood oxygen saturation. So, despite, xylazine showing that xylazine does worsen the process of inhalation, we showed that xylazine really profoundly decreases heart rate. And so at the end of the day, I think that the effects on the cardiovascular system might end up being as important, if not more important, than the changes on breathing. Just from an idea that, fentanyl is going to lower the oxygen saturation of the blood. And then if xylazine is going to lower the heart rate, we can, there's less total oxygen reaching the heart in a given amount of time. And so, so it's possible that that's the mechanism for the increased lethality in mice.
It is fascinating and scary at the same time to be, one, doing these experiments, but at the same time thinking about that these reactions happen in humans on a daily basis, because the fentanyl is cut with xylazine.
Mm-hmm. Yes.
You mentioned before we hit record that you're affiliated or you're working with an organization that allows you to, that tests these drugs, that people just come in and they get tested. I'd love to hear more a little bit about that. But in the context of your work, I was curious to see if whenever you were doing these experiments, did you vary the quantities or the ratio of fentanyl versus xylazine, and did you observe anything that's worth mentioning? Like, does more xylazine is more xylazine worse in combination with fentanyl or less xylazine? How much do you need for it to be lethal?
Yes. Excellent question. So we didn't do lethality studies here, and it turns out in the literature the lethal dose is very strain dependent. So I won't really speak to that here, but yes, we did vary the doses, especially the doses of xylazine. It's really hard right now to find data on the sample ratio of fentanyl to xylazine, like in the actual samples that people are getting. The organization that I'll talk about in a minute uses IR spectroscopy and also sends out, but they just get back major, minor, constituent, not any sort of percentages. So I chose the ratios of fentanyl and xylazine to use based on postmortem toxicology in humans, where you can see, which of course is not a perfect readout at all of what was in the original sample, but it's just the best that we have to go on right now. And so from these data, it seems that there's anywhere from a 1-to-1 to a 1-to-10 ratio of fentanyl to xylazine. And so, I used accordingly a 1-to-1 and a 1-to-10 ratio. And we pretty much see similar results in that, at almost all of the doses, at most of the doses tested of xylazine, that, the combination of fentanyl and xylazine, again, mirrored that of xylazine alone. So it seemed to, drive the effect when the two were combined. But yeah, I think it's certainly, it's an interesting point too, because some people have wanted to, claim or have wondered whether the total, the average quantity of fentanyl in these illicit samples is actually maybe a little bit less in those that are cut with xylazine, from the standpoint that maybe dealers are trying to, make their fentanyl last a little longer by adding the xylazine. And so that I think is something really interesting that the field wants to get a clear, understanding of. But as of right now, we don't have a complete understanding on that. I think that would be really relevant to know as well. But to take a second to touch on RED Project. Thank you for asking more about that. We have a relationship with an organization in Grand Rapids, Michigan, and I think this is a really phenomenal place. They started off as, they're one of the biggest safe needle and safe use kit distributors in, I think, all of the state, maybe even all of the country. And recently they made an agreement with the local police to be able to start a drug checking service. And so, people bring in whatever they get from their dealer, and, they get an IR spectra on the spot. They don't have to wait for that. They get the results immediately of what's in it, no idea what ratios. Maybe they could get a little idea, but, really just here's what's in it and potentially in this order. And then they also send out, a sample for further testing at UNC, and they provide, like a semi-quantitative readout of the samples. But, I think this has been really fantastic for us, because we get a readout of drug use in Michigan right now. And so that was what had inspired the Xylazine project. Was they've seen a 30 to 60% increase in the number of fentanyl samples that contain xylazine in the past year. Actually, now it's probably been a year and a half, but I mean, that's, a really substantial increase. It's not like even doubling from 1% to 2%, it's 30 to 60%. I mean, that's pretty insane. And it seems to be an issue concentrated more on the eastern part of the country and, the South as well, but the Western states as of now are seeing less xylazine adulteration. So, what that kind of says to us is that this is something that's happening on the local dealer level. This is the fentanyl that's coming through Mexico is likely fentanyl. And then at a more local level, dealers are cutting it with xylazine, since we're only seeing the trend in this part of the country.
It's incredible. I can only think about, the series Breaking Bad, when Mr. White, made the purest crystal meth, thinking, I think it wasn't even him. It was the other guy. I can't remember the name of the character, who said, well, if people are to consume drugs, I'd rather have them something pure and not cut with anything. But I think it's an epidemic in general, and it's so sad. And unfortunately, it happens, but at the same time, we're, people like you and these organizations are here to get a sense of what's going on, and can we develop drugs, medications, not
no, I
drugs.
I know what you mean.
Oh, there's a nuance there to help people and beat these addictive behaviors, and it's a very difficult thing to go through.
Yes, it really is. And one last thing on this topic, and then we're free to talk about something else. But something that I think is interesting and perhaps gives like a smidge of hope for the xylazine situation is that 90% of people using illicit fentanyl actually don't want xylazine in their samples. People who are aware of this potential adulteration don't want it. It's not viewed as a positive or as a plus, and that's largely because xylazine induces skin ulcers. These really, nasty skin ulcers, or abscesses almost in people who use it IV. Not in everybody, but in a large enough percentage. And this is mediated through alpha 2B activation in the periphery, causing vasoconstriction. And so essentially just the lower blood flow to the surface tissues, makes it easy to get infections, which then are really hard to heal. And the kind of scary part is that, they can happen anywhere on your body. Some sorts of injection wounds, right, are only local site of delivery, but these, because it's just the vasoconstrictive aspect, can appear anywhere. And so, I think it's important to mention and to understand that, this is a dealer driven choice. This is not, driven by the people who certainly want to be using their fentanyl, but they would prefer it, as you just said, to be pure. They would prefer it without the xylazine. So, hopefully, I think that kind of gives a way for places like Red Project to make a huge difference, because, people can they go to Red Project, they know they just got it from a certain dealer, and then they find out, okay, it's fentanyl and xylazine. Okay, maybe I don't want to go back to him or her. Maybe I want to go to somebody else who, gave me a sample that didn't have xylazine in it. And so, it's possible that in that way, this making these informed decisions could have some effect on a street level. It's just, like you said, it's a really difficult situation for the people who have these addictions. And, I'm happy at least that there's somewhere that they can go to, at least be informed of what is in, what they're putting in their body. I think
that's unfortunate. How does this work that we just talked about fit into the grand scheme of your PhD thesis? And maybe we can end, because I know we're at time. I didn't ask you if you had a hard stop at the top of the hour, but maybe we can end on what next after your PhD.
Okay, fantastic. So the xylazine work is going to be my second chapter of my thesis. The first chapter was delving into furanol fentanyl, which was a really popular fentanyl analog. It was like the second most trafficked analog within the United States in 2019. It has kind of faded out because essentially prior to 2018, if you just made any little modification on the fentanyl scaffold, boom, you were in the clear until, the DEA got a hold of your particular compound and then, made it illegal. But until then, you were theoretically dealing something that was legal. And so, an abundance of analogs popped up all around the country and the world. And furanol fentanyl was among the most popular. In 2018, they passed this blanket law that anything on the fentanyl scaffold was automatically illegal. And so, as you can imagine, most people just went back to making regular fentanyl because, it's easier, et cetera. So the analogs, for the most part, have faded out of sight. There's some really scary ones still floating around, like carfentanil, which is actually, used to tranquilize elephants. And so, it's even more potent than fentanyl. It's just terrifying to think about. But for the most part, the analogs have kind of faded out. But I did a really thorough look at, furanol fentanyl in comparison to fentanyl, and then also to morphine, which is, on a different scaffold than fentanyl and has a lot of different properties there as well. And so this, I did a lot of in vitro and in vivo characterizations, looking at everything from binding affinity to downstream, responses, and then looking at, again, the same metrics or the same techniques for respiratory depression as in the xylazine paper, using like whole body plasmography and pulse oximetry, and then also doing anti-nociception assays as well. So that one, I think it's a cool, thorough work, but less relevant, less topical than the xylazine, which is why we pushed to get that one out first, because I think it's more useful for the world right now. And then my final body of work that I'm planning to do for my thesis is actually looking at molecular signaling in fentanyl-induced respiratory depression in vivo. So I'm really excited to learn stereotaxic surgery, and I'm going to be injecting, I have some floxed mice arriving, and then I'm going to do some stereotaxic injections to knock out specific molecules downstream of the opioid receptor in the two brainstem regions that are thought to mediate opioid-induced respiratory depression. And so I'm excited for that. I think that'll be an opportunity for me to learn a lot of new techniques and hopefully contribute some meaningful knowledge to the field. Of course, it's possible that the targets I've chosen won't have, an effect on respiratory depression, but that's okay. Negative data is still data. So that's what I'm kind of gearing up for now is that last leg. And then post PhD, I want to stay in academia, which is kind of rare for people at my stage right now, at least in pharmacology. I think, pharmacology opens up a lot of industry doors. And so I would say like 9 out of 10 people that are, getting their PhD right now in pharmacology, maybe 8 out of 10, want to end up in industry eventually. But I really like academia. You know, I really want the freedom to keep studying what interests me specifically. And I've just really enjoyed the flexibility and the focus on like learning, you know, that is in academia. So I would like to postdoc in human opioid research, I think. So this spring, I'm doing a sabbatical with Mark Greenwald at Wayne State University. He is there. He's the associate chair for research in the psychiatry department. And then he also is the director for their substance addiction research division. And so I'm going to get to work with him for, you know, about 10 weeks, looking at polysubstance use in humans and how that, you know, affects different neurocognitive and affective traits. And so I'm really excited about that. I think that'll give me like a good idea of whether I like the human research aspect or not. But as of right now, that my plan would be to hopefully pursue a postdoc where I can, you know, work with human patients and you know, maybe eventually have my own lab that has some marrying of the two, you know, the preclinical and the human patient populations and data. But that's the loose plan right now.
I love it. I love it. All right. I have two last questions for you. Number one, what's your favorite GPCR? Like, I don't know the answer to that.
Oh, you know what? I'm so glad you asked at the end. It's actually DRD2.
Okay. I was sure you're gonna. I was sure you're gonna say opioid receptors, mu-opioid.
Right. Well, I if you asked me that in the beginning, I was going to answer honestly, but then say for the sake of the episode, why don't we pretend it's the mu-opioid receptor? But you know, no, I really love dopamine. Love the feelings that dopamine bestows, and so for that reason, I think my true favorite GPCR is DRD2.
Awesome. I love it. I'm taking notes because I have an Excel sheet, but my team will get back to me and will say, hey, why didn't you take this? And I'm kidding. Obviously, they won't say that, but I try to make their life as easy as possible. All right. What should we title this episode? That's a tough one. And I leave this typically at the end because it's not an easy answer.
Sure. And I think, you know, partially would depend on like how the session goes, I imagine.
Yes. I think it's more, this is the opportunity in the episodes to be as creative as we want to, and maybe, you know, step away from our, let's say, scientific box and try and think about, okay, what did the episode or the conversation leave you with as a feeling?
Okay. I think, you know, we've spent a lot of the talk, you know, kind of going through, you know, the different, I guess, the trajectory that, you know, took me here today. So maybe we could title it something along the lines of, or something to do with, you know, a non-canonical path to a PhD, or something like, maybe not non-canonical. That's probably too jargony.
That's okay. I think people are, you know, we need something catchy. So for example, I've had someone suggest for a previous guest computational chemistry of olfactory GPCRs, which I thought was very interesting. Or there was one guest we had, and we called it research the gene that's fifth generation. Just because the guest and their grandfather were scientists, but not not. So we can be as creative as we want to.
I feel like I would also like somehow, and I don't know how to merge these two into one title, but I would like to also, you know, maybe have in the title something about the fentanyl and xylazine epidemic, just so that people who are interested in either aspect would maybe know to listen to the episode. So what if we did something like, I don't know, a non-canonical journey to academia, and then I don't know if we would do like and the xylazine. I'm not sure how to merge these two.
So what I put down is non-canonical path to a PhD via the xylazine and fentanyl epidemic or opioid epidemic.
Okay, yeah, I like that. Why don't we just put via the opioid epidemic, maybe, because the xylazine, I feel like wasn't quite okay. I love it.
Awesome. To a PhD. Awesome. I think we did a great job on that one. I love it too.
Okay, awesome. Well, this has been so much fun. Thank you so much for hosting me today.
Absolutely, Catherine. Don't go anywhere. I'm gonna stop recording, and I want to get your honest impression, non-recorded impressions of how did it go. All right. Thank you, Catherine.
Thank you.
Huge thanks to Catherine Demery for joining us today. Her journey is a powerful reminder that science is not always linear, but when driven by purpose, it can have a profound impact. If you enjoyed this conversation, subscribe to the Dr. GPCR podcast and share it with a colleague. Leave us a 5-star review on Apple Podcasts and subscribe to our channel on YouTube. Don't miss out this week's lesson in Terry's Corner, where Dr. Terry Kinakin breaks down competitive versus non-competitive antagonism, teaching you how to read dose response curve patterns and avoid misconceptions in receptor pharmacology. Most importantly, take the next step in your own journey. Become a Dr. GPCR Premium member today. You'll get, among other things, our exclusive weekly news, curated jobs and publications, and more than 4,000 minutes of expert-led courses designed to keep you at the cutting edge of GPCR pharmacology. As always, thank you to the entire Dr. GPCR team for making this episode possible. And of course, a special thanks to our partner Silteris for supporting us and accelerating GPCR drug discovery. Because receptors don't work alone, and neither should you. Until next time, stay safe.